algae, and you can use it as food for your rotifers. But be careful for contamination due to parasites or other problems. Now, why do we need so many rotifers? You can see in this slide that one single fish larvae needs at least 100,000 rotifers during the first 2 weeks of its life. So this is the point when the fish larvae hatch, you need to have extremely high density of rotifers. The species which are most commonly used for aquaculture are Brachyunus plicatilis, Brachyunus rotundiformis, Brachyunus demediatus, Brachyunus caliciflorus. These are the commonly used taxa for marine and freshwater systems, and try to culture them in high densities, but please plan your cultures, because there is no point in culturing 1,000 liters of rotifers if you don't have any fish larvae. They will grow and then they will die. So depending upon when you need these rotifers, invest in the culture. The nutritional value is very high. You can see over here, Artemia is very expensive, but Moina, which is practically free, has better nutritional quality because it has no ash. Similar is the case with copepod. So this, if you develop this culture technique, if you learn it, it is extremely useful. Consider the nutritional value of rotifers with different diets, with yeast and with chlorella. You can see that the protein content of the rotifers goes down on a diet of yeast. So yeast can be used to maintain the species, but eventually you should feed them algae before giving them to the fish. What are the problems that we can face in cultures? You can have different types of contamination due to ciliates, other types of protozoans. You can have problems due to predatory rotifers such as Asplankna, or due to other predatory organisms such as flatworms. But if you keep your cultures clean, if you change them with in a regular basis, and if you are careful and keep observing the cultures, you will not have these problems. And if you do, you should be able to separate these predatory organisms or the others through meshes. Otherwise, you have to begin the culture all over again. So this is the Asplankna, which is a predatory rotifer. It can be quite problematic. So be careful, because if it enters a culture of Brachyunus calciflorus, within 2 days, you will only have Asplankna. Asplankna is jelly-like rotifer. It does not very good food for the fish. So this is not very useful for you. Thank you very, very much for your attention. And I'll be happy to answer any questions that you may have. I know that this talk was very fast, but it was just to give you an idea about how rotifers can be cultured. And you need to practice. And while practicing, if you have any problems, please do not hesitate to write to us, and we will be happy to answer you. Any questions? Or maybe you can start the session with Dr. Sarma and ask any questions there also.
Good afternoon, everyone. There is a question.
Yes.
Madam, how long this culture can be made viable in laboratory condition? One thing. Secondly, all these zooplanktons, they are herbivorous, or they may feed upon other smaller zooplanktons in culture.
All of them are not herbivorous. There are some which are carnivorous, and then you have to offer their prey also. So you have to culture the species of interest plus the prey. And for the second part of your question, how long you can maintain it? It depends how carefully you maintain the organisms. For instance, we have one species of copepod which has been in our lab for 19 years now. And we have some rotifer species that have been there for several months. So it's not difficult to maintain them. For instance, we have 4 years, 5 years. It depends upon how you maintain these organisms.
Suppose
It's not. Mhm,
we want to maintain it in laboratory condition,
mhm.
so we have to supply the algal food regularly or any other thing to be required to be
No,
maintained.
you have to change the culture. You have to change it using a small mesh, put fresh water and fresh algae. Because if you do not change the culture, it's called changing a culture. That means that you take the culture, you filter it, you pass to a fresh medium of algae and water. Because if you keep it in the same medium, it will develop, it will accumulate all the toxins. So that has to be done in the case of rotifers at least once every alternate day, at least, if not every day.
Is it possible to make their monoculture?
Yes, yes. We have monocultures. You start with one single individual and it's known as a clone as long as males do not appear. Otherwise, it's a monoculture. Yes, it's very easy to do.
Okay, thank you, madam.
Thank you very much for your questions.
Any other question, please? Good morning, ma'am.
Good morning.
Ma'am, can we use the same rotifers for all the carp species of fish?
Yes, yes. You normally you can use Brachynos caliciflorus for freshwater species and Brachynos plicatilus for marine species of organisms. And you can use the same species for all the fish. There's no problem about that.
Or it is changed according to
No,
the
don't change the species because if you are interested in fish, don't change the rotifer species because then you will have contamination with other rotifer species and then you will have problems of competition and exclusion and all these things will also come up. So use one rotifer species.
Thank you, ma'am.
Thank you.
So, a heartfelt thanks to Dr. Nandini Sharma ma'am for her excellent lecture on species diversity of rotifers as well as plankton culture techniques and collection of samples, concepts, and various calculations. And of course, the interaction session proved the interest created among the audience. And thank you so much, ma'am, on behalf of everyone present here for this wonderful presentation. Thank you so much.
Thank you very much. Thank you.
There is a break of 15 minutes. So exactly after 15 minutes, please be here so that
Yeah,
we can continue.
because that is a very important and a very interesting session with Dr. Sarma. He will give you very valuable tips. Do not miss it. Come back.
Yes, ma'am. Thank you.
Thank you so much. Thank you.
Chalo, be here within 15 minutes. Okay.
Thank
12:30,
you.
everybody should be there. Okay, thank you, ma'am.
Thank you.
Recording stopped. Recording in progress.
Good afternoon to everyone. We start after the break this session, and I am going to introduce as Sharma sir. Sharma sir is an aquatic biologist known for his research contribution in the field of freshwater zooplankton. He is full-time professor of the National Autonomous University of Mexico since 1995. He is elected member of Mexican Academy of Science, International Society of Immunology, Royal Society of Biology, London, and National Researcher of He has published more. He has, pub- he has published more than 300 articles and book chapters of his 285 scientific works are from Asia Index Journal, including Nature. And he is currently editor of Rotifers News Virtual Newsletter. Today he will discuss about the topic of identification of rotifers from different sites. Then I welcome to Sharma sir to deliver this topic. Thank you so much.
Thank you very much, Madam, for your kind words. I will start a very brief description of what features should be considered for rotifer identification. For this, I will share the screen. Can you see the screen?
Yes, sir.
Can you hear me as well, right? Okay. So first thing is very important to identify the species. Before you do any work, it could be ecology, it could be ecotoxicology, aquaculture, for any purpose, any species that you are culturing, you should know what species you are culturing. So there are experts. So when you hand over the species, they will identify for you. However, that is not sufficient because you need to know also what species you are culturing because it is your own species under consideration. So what you have to do is first thing is
Sir, please share the slide mode, slide show.
it's not in the slideshow. It is in my okay. I'll put again. Um, it is still not in the slideshow. Um, yeah. Can you see the full screen? If not, I will stop and again share. Okay. Share the screen. No. Is it okay now?
Not yet, please.
But my screen shows it is in full screen. But
Okay.
okay. Can you see it? Okay.
Continue.
Okay, so the first step is rotifer collection. So how are you going to collect the rotifers? So you need to collect the rotifers using 40 or 50 micrometer mesh. You can also use a little bit larger, but smaller species like Anurops fissa will you will not be able to collect, especially for quantitative analysis. There are species which are as small as 40 micrometer or even smaller in width-wise. So it's better to use 40 to 50 micrometer mesh is sufficient for you will collect about 95% of the rotifer species in a given water body. As I explained yesterday, you can collect repeatedly in different parts of the water body and also in during different seasons to have very high diversity. Once you have the collections, now observe the sample first
under the microscope, live samples under the microscope. Why? It is important to characterize, to detect certain features. For example, the corona structure, the foot length, the toe length, the movement. All these factors are important to determine the species. Therefore, you need to have live cultures, for live specimens to observe for some time before you fix them. Well, in most cases, we do not bring the samples to the laboratory, live samples, because it is difficult to transport. So you fix them there and then bring them to the laboratory. Or in case you are interested in, if you are interested in some background noises are coming, I don't know from where. In case you are interested to study molecular biology of the species in question, well, you can fix them in the laboratory, you can fix them in the field with alcohol, 99% alcohol. The general idea is that you have to use the cold alcohol to detect maximum, to collect maximum DNA content. Now, once you have sample fixed in the alcohol, but do not forget to fix sample also in formalin, because in order to identify the rotifer species, formalin fixation is very important, because formalin will give the exact structure of the lorica or the body of the rotifers and the spine lengths and so on, because these are important for determination of, for detecting the identifying, confirming the species status of different taxa. Now, for certain species, not definitely for Brachionus and Caratella, for other genera, you need to separate the trophy, especially the members of the Notomatidae as a family. All of them need trophy separation. Trophy is about 25 micrometer in size on an average, and is difficult to separate the trophy without adding either sodium hypochlorite solution. This is a commercial grade. You can also get 40% solution, but this is carcinogenic. Generally, industry does not supply unless the demand is specific for a particular purpose. But otherwise, 78% this is normally used in the house for cleaning the floors and all. This sufficient to add. What you need to do is collect one individual, only one individual in one slide with a small quantity of water, but keep it under the microscope continuously. Usually between 10 times, 10 into 10 is obviously, or even 20 to 10 if you have this kind of magnification. Uh, Compound microscopes are necessary for this. Um, Stereo microscopes do not have that much resolution. So it is necessary to keep the specimen under the microscope, live or fixed material, add 1 drop of sodium hypochlorite solution. It will dissolve all the muscles, and what is left over is simply the structure of simply different parts of the trophy. These are hard parts. They have also a small segment of chitin, and they do not get dissolved easily with this concentration of sodium hypochlorite. Once you have this trophy under the microscope, you can observe them carefully and draw the diagrams. For drawing the diagrams, it is important if you have microscope fitted with the camera lucida. If not, no problem. Take regular photos with normal um, cameras. You can also use your cell phones. Cell phone camera also is really useful for taking the photos. Okay. So this trophy separation is important for several genera of rotifers. Now, if you want to study the corona structure, how it is, some of the slides I showed you yesterday, that the corona is very widely expanded. To get the expanded position of the corona, Edmondson suggested to use hot water. So you collect zooplankton in a small petri dish, and then boiling water pour suddenly on the top. So what will happen? Those that receive the hot water, the muscles immediately expand and the cilia get expanded. Then this will be useful for identification of some species, especially if you are interested in the corona structure. Now, electron microscope is not needed in most cases. But if you have, it will have a better resolution, obviously. It is as a rule, for zooplankton guides, they are not based on the characters that are not visible by light microscope. It means they, for identification of the keys, zooplankton or even rotifers are identified using the keys. The standard keys are produced by observing the characteristics that can be visible only under the light microscope. If a feature is not visible under light microscope, this is not useful for identification of the for the construction of the species keys. So this is also important. But what we need to study in order to identify the species, first of all, external morphology. How small or how big is the animal, and whether it is rounded, spherical, it has got spines. If spines are there, how many spines are there, both anterior and posterior, and dorsal and ventral plate structures. Dorsal plate, if it has got any ornamentation, it is very important for Lecane, the genus Lecane, and other species also. Uh, Dorsal and ventral plates are separated by a membrane. In order to detect this structure, you need to rotate the animal in different orientations that to get the cross section. For example, Lepidella, Euclanis, all these are identified based on the width of the animal. That is how the dorsal plate and ventral plate are separated by a membrane. How thick is the animal? So for that cross section is needed. It using a dissection needle under the light microscope. You can move the animal um, uh, laterally, or you can move the dorso-ventrally. Then you will detect uh, the the structure of the dorsal plate and the structure of the ventral plate. Now, trophy morphology, in some cases, the trophy is asymmetrical. So some parts will fall out very easily. Therefore, when you add sodium hypochlorite, it's better to observe immediately. Once you lose the sight of the the trophy, you cannot detect it again because very, very tiny, and the light microscope is not sufficient to detect. But if you follow one individual adding the sodium hypochlorite, you can definitely detect the trophy, and as fast as possible take photographs or draw the camera diagrams. Now, what are the literature? What kind of literature we have now? We have the standard literature by Coste. There are two volumes, and this gives you these two volumes are essential to identify. But this does not cover the group Deloidia. There is another book separately. It was produced by Donner, Joseph S. Donner in 1968, 65. So that is useful for identification of the Deloitte rotifers. For all monogonal rotifers, Coste is, although it is, the title says for Central Europe, but it's for the whole world. It includes also species from North America, and about 1,500 species are included. That is more than sufficient. Recently, because the coast volumes become outdated, especially for certain genera like Brachionas, Costa is not useful because he clubbed many species into some kind of variety or forms and so on, infra-specific taxa. So modern studies indicate that it is necessary to elevate them to species level. Therefore, Costa and Schill modified brought out some series of articles, but most of them are included now in the special SPP academic publishers. There are series of volumes. There are about 14, 15 volumes on not only on Rotifera and many other groups, the Claudosera, the Copiopoda. They all have appeared, and some of them are still in process, but probably they may not see light because the chief editor in chief is Professor Henry A. Dumont. He has become a little less active now, and he has also some problems with his eyes. And authors are also not willing to prepare the volumes in relatively faster. Even the already published works need upgradation. that is also not done for some species, especially molecular tools have given more species. Fortunately for us, for many groups, including Brachyonus, Plicatlus alone, which is supposed to be only two species, the species complex of two species. But now we know there are at least 16 species are detected based on the molecular tools. So some of these aspects are not included in the older guides, yet these guides are useful to a great extent unless you are interested in subspecies level, these guides are sufficient. Now, for literature within India, B.K. Sharma's nearly 200 articles, mostly on taxonomy of rotifera. A few are also on the taxonomy of the of Cladocera, but the majority of them are on taxonomy of rotifera. You can consult them. Many of the articles are published in Zoological Survey of India, monographs, and some of the monographs are exhausted already. So you can depend on this. Do not directly go to Coste because you will be misled by seeing the number of subspecies, infraspecific forms, and so on. And many of them probably exist only in Europe. So B.K. Sharma's guides are useful, and there are also other authors who have produced, like Dhanapati also made some guide, identification guides. So some of these things that I'm mentioning, these can be done even in an undergraduate college. You do not need university facilities. You do not need the expensive equipment, projects, and so on. Whatever projects given to you by the UGC and CSAR, I don't know, some such organizations, the smaller budget is sufficient to develop this. One of the greatest taxonomists in the copiopoda, for example, calanoids, is unfortunately passed away, Dr. Y. Rangareddy, whom I know for the last, I know him at least 40 years, and then unfortunately passed away, but his guides are still useful. Even researchers from Europe and North America are dependent on his calanoid identification guides. So that is the power of doing the taxonomical works uh, using simple equipment. For example, camera lens is more than 150 years old model, and some of them are updated, but still the concept is the same. So these can be easily prepared in the time that you have after teaching, for example, some depending on the workload that you have. All teachers have teaching workload obligation, is independent, whether you work in an undergraduate institution or at the university. We still have to give classes, and then that takes a lot of time, but that also gives you an opportunity to interact with the students, and we explain what cannot be generally published formally in journals, articles, and so on, but you can discuss with them and transfer the information. Some of these techniques you don't find them in any single book, but they are all by experience or by reading literature part by part from different journals. You get this information. So this is one part I would like to tell you. The second part I would like to show you is this. I will stop and take another one. Uh, and I share the screen for another uh, aspect. Okay, this is not needed. Let's see. Then I will go for the uh, 2 volumes of Coste. Dr. Nandini already mentioned about the volumes of Coste. We have some copies of that one, but uh, the real description is like this. Okay. Now I will share the screen again.
Yes, okay. Yes, we can see the slides.
You can see it?
Yeah,
Yeah,
yeah, it's okay.
excellent. So there are 2 volumes of Rotatoria. Old name is Rotatoria, recent name is Rotifera. It's a Central Europe, it is there are two volumes. One volume is exclusively in the text. Another volume is simply the line diagrams. No photographs are used, so you can also consult them. The books, each volume is about $100, not really expensive. And then you can also purchase some in Amazon suppliers also used books there, but they are really good. Some of the retired rotiphorologists, so they don't find any other person to purchase, so they sell online and they cost about $50. And some of our students purchased them. I don't know they're still available, but the original cost of each of them is about $100 each volume. And it's worth. And we scan them because we use regularly in the class, because this is the single monograph available today, comprising of more than 1,500 species of monogonons. So that is more than adequate for any level of teaching or research, uh, unless you are more interested in subspecies level. But this book is worth to have it. Now, next part I will share. Are you able to see the figures? Okay. So these meticulously drawn these figures with only camera lucida, and these all you will see. This is the scanned version I use for the laboratory. It's in the PDF version, and on the right side you have the photos, and the left side you have the text. So the text I just copied and pasted just below the figures. All figures are drawn with simple camera lucida, not any expensive camera lucida are available attached to the microscope. Even monocular microscopes can also be useful for this, and they are relatively cheap. The prism type, the mirror type, the tube type, there are different types depending on the cost. But even the primitive ones are sufficient to draw these diagrams. All of them are drawn with pencil, and then they use India ink, China ink, whatever it is, and then they are all drawn to the same size. So this have Poste, this from Poste, only textbook. The part of the description, um, I have not scanned here, I have not brought it here, but this is useful for identification of all the species that you have in the world. At least vast majority you can get from this. Okay, now Dr. Nandini already also mentioned about uh, Hendrik Seger's works, so I will share that as well. This work I will give you, Dr. Nandini will send that to you. Uh, this is share the screen again. Okay, this is Zootaxa. Uh, they have issue number 1564 and annotated checklist of rotifer. This is most authentic list of species. Whenever you are writing a species, the descriptor or the author of the taxon, you will find here completely. This is the how zoogeographical distribution of the species. Um, this is used by Kuhn Martens, and then many people use for the zooplankton. This is an excellent model. All the zoogeographic regions are given here, and the species are distributed. Some are restricted to Nearctic, some of them restricted to Palearctic, some of them are Neotropical, Afrotropical, and so on. So the importance of this one is, as Dr. Nandini already mentioned, the correct species name, taxonomic stability, the author, and the synonyms of all the species of rotifers, including the Deloids. So this is a very valuable contribution. I think Dr. Hendrik took to write this one at least 15 years to collect all the species and synonymize many of them. Um, this is how the species, a genus, the valid species under its name are listed. And here you will also find the location where you will find them. If a species that is not found in India, for example, is found only in Europe, you should first think, consult this one. Why you find the species in India, and you can, I don't know, some kind of background noise is coming. Okay. Anyway, this is how you identify all the species. You can confirm the taxonomic status of all the species. It is approved by the world taxonomists and is being used widely. For example, if you have one species, the Neuropsicoilata, and there is also Suzuki made several subspecies, and they are all synonymized here based on the observations and literature figures supported by other researchers. This is a free copy you can download easily from the internet, from the is open access. Okay. Now, the another aspect that I have received from Professor Das, some of the figures, how we identify them. So I will see again share the screen with you. Uh, let me see where it is. Um, okay. Can you see the species? So species of this kind are difficult to identify, but by experience, I can tell you what species it is. It is the genus Lacane, species Lacane Clostrocerca. What is missing here, or what is not in the focus, is out of focus, is the toe. There is a toe, distinct toe here. What you are seeing here is the anterior part of the animal, that is the trophy and corona. Somewhere trophy is located here. This is the corona part. I'll show you. Uh, no, this I cannot see this one, but anyway, this is the corona part has come out. So you need to add high concentration of formalin. So as I mentioned before, that 4% formalin, normally the formalin, commercial formalin is about 33%, 34%. Calculate in order to get the 4% formalin at the end, the concentration of the formalin in the sample should be 4% or even slightly lower, no problem. For lasting for more, usually you can use 5% as well. So if it is not properly, if the concentration of the formalin is not sufficient, uh, then the animal gets expanded irregularly and you cannot identify the species. So this is one of the problems we have in when you are fixing the sample. Make sure that you add enough concentration of formalin. Then I will show you some of the figures again. Again, I have to share because I did not put all of them in the last night only. I received them, so I did not put them in the PowerPoint presentation. But anyway, I can show you some of them. Um, uh, okay, so again, I share the screen. Okay, I think you can see this. It's the same species. Um, But again, another view, I still do not see the toe. The foot and toes are located. This is the foot one segment. This is the toe. Foot in Lacane is one segmented. So I don't know why it is not moving to the next level. Um, okay. Yeah, it is moving. So this is Deloid rotifer. It's a Philodina. The genus is Philodina. There are many species in Philodina. Philodinidae has about 108 species, so very difficult to identify them unless you have the live individual. Costae, the volume Costae does not include this group of animals. So for that, you have to consult the other literature. Kutikova also made recently about 8 years ago, one volume, or you can consult also Donors special volume of 1965, exclusively order Deloidia. In those days, they consider this as an order. That can be identifiable only with the live species and how corona is expanded. If corona is corona, how it is expanded, and foot, toes, spurs, if there is any such structure. This is the same species in another view, also the same species. This is what we are saying. The corona is bilobed. There are two lobes in the corona. That is useful. Here I can see one part of the corona, and this is another part of the corona. It's out of focus, but you can. There are programs, by the way. You can merge them, the different photographs, into a single, like stacking they call, and that will be useful to represent all the features in a single slide. And this is another one. This is again is a species of Lacane, uh, possibly is also what is you can see as a very small structure is foot. The toe is located here. This again, Deloidia, the Philodina. This is Deloid. When you add formalin, they get contracted irregularly. That is why formalin is not useful for identification. Fixed species of delights are not identifiable because the corona is withdrawn, the foot and toes are withdrawn, the spurs are withdrawn. So it is just all the delights, different species of delights will look more or less like this. So it is not easy to identify them. Therefore, this is something you need to observe only in under the live conditions. This is another view of the same. This is this is Lepidella. You will see Lepidella has a three segmented foot. There are three or four in some cases foot, and what you are seeing is a toes. There are two toes well separated, but in this case they are joined. But if you are using 40 times magnification, you will definitely see the two clear toes. In some cases, some individuals, uh, the toes are very clearly separated. So there is an anterior end. This is head opening is lunate, something in the form of half moon, and there is a ventral plate. And a ventral plate, you also have another opening, ventral opening. The foot opening is also semicircular. Usually is a long. So the number of segments are constant. The foot pseudo segments, they are called, they are constant for most genera. What is more important here is variable is the toe length. The toe can be length, and in this case, as I mentioned before, cross section is important. That you need to orient the animal. How thick is the animal? The dorsal plate and ventral plate, how they are. Uh, uh, this is Lepidella patella, Lepidella ovalis look alike. The only difference is the width of the animal. Therefore, if the width is not given, it is not easy to identify. More or less, we can say is a Lepidella ovalis, but it is necessary to check the cross section. This is another species. It is the form you can see uh, more or less like a barrel. It's like gun barrel type. Is a very long. Anterior part is wide, and the number of spines here, trichotria, trichocerca, then all these species. This is another genus. So altogether, we need to see how many spines are present on the part of the posterior, and so I would
This is
say that.
Mytilena, no?
Yeah, I'm coming about Mytilena.
Mm.
Mytilena ventralis has got two spines, additional spines here on the ventral part. This is the dorsal part. Ventral part, there are two more are present in Mytilena. Mytilena ventralis is very common. Because the spines are absent here, I would say it is Mytilena ventralis. So, as I mentioned, Trichocerca, species of Trichocerca, they look very similar, but the difference is they also look very elongated, but they also got foot and toes, but this kind of strong spines are present only in the anterior part. So you get more or less confusion when you are checking many different varieties, genera in the same water body. So you need to have first first and look at the all the figures of rotifers, so you have an idea how they look like. This is I something went wrong again. I'll check again. Um, this is Lepidella species of looks like a Lepidella. Um,
1 minute.
Something has gone again. I'll put, please 1 second, because this is gone out. 1 second. I'm trying to see this species. That was this one. No, this is we have already seen. But I'm opening with another program that is why I'm not able to get this ID. This one. Aha, this is the one. Lepidella triptera. Lepidella triptera. So I didn't stop the screen and again share the screen. Lepidella triptera. Can you see the animal Lepidella triptera? Here you will see, when you see the animal here, you see some structure. I can see some kind of elevation. So this is Lepidella triptera. So in the poster you get this information. Many different morphological forms are there. These are not different species, but different morphological forms are there. So this also you can get in poster, but you need to draw the diagrams, because in order to see how variability in the morphology can exist in this group of organisms. Any other species that I have? 1 second, let me just stop the screen. And I think these are the only species that I have from the list that I received. But if you have a species, please project on the screen. I can see them. I can try to identify them. Two species of Mytilena that you have, Mytilena ventralis, Mytilena mucronata, both are present in India. So you can get both of them. Mytilena acanthophora is also there, also common, coexist in the same tropical conditions, but it's mostly associated with the aquatic vegetation. So it is important to identify the species in order to use them for as indicators of water quality, or as bioassay organism, or even culturing as live species.
Excuse me, sir. Hello.
Sir,
Yes.
Am I audible?
Yeah, yeah.
Sir, we have sent you one more photo of the rotifer species that we observed, the last one.
One, did you send to my email?
Yes, sir. Yes, sir. Sir, please cross check.
Yeah, I'll just check it.
Yes, thank you.
if you take nice photographs like these with your cell phone and send it to Dr. Sarma or send it to me, we can try to help you to identify them. Dr. Sarma is the final word, of course. But after that, we can also interact with other people in the world and try to help you. So please don't hesitate, especially the young people. Please send us.
Yes, ma'am. Thank you so much.
Welcome.
I did not get anything here. I downloaded all the photos.
Which mail did you send it to? Did you
Sir,
get it?
the last one in the photographs I have
Okay.
sent.
Okay, then let me see. Yes, you're right. Yeah, okay. Yeah. I will share now the screen. Correct. So this one is, then in that case, I have to show again, share the screen, and this one. Is this one? Yeah. This is Brachionus angularis with a resting egg. You will see some part is darker the other part is transparent, and it is a resting egg of Brachionus angularis. But it is with still corona was expanded and formalin concentration was not sufficient, or it is the dead individuals in the field and you added formalin. Dead individuals who generally have this kind of trophic corona coming out of the lorica. So, but I can tell you that what species it is definitely Brachionus angularis because based on the egg structure. But I still need to see if there are any spines. There must be only two in the median spines. The size very small and two must be there. Then then it can be confirmed much better. But as of now, I can say it is Brachionus angularis. But you still need to see the anterior spines. So you need to take photographs in different orientations. Just do not put only one orientation. Second important point is not every individual in the zooplankton collection can be identified because some of them do not have the proper orientation or they are fixed when they are dead already in the field. So morphological variations can confuse us. Therefore, you need to study more than one individual. In other words, some kind of study of a population. It also gives you the idea of the variation, the spine length, spine form, also the body size. So body size is also important. You need to put a scale to know how big is this animal. Angular is generally very small. It is about less than 100 micrometers, about 80 to 90, whereas Brachionus calciflorus is very big, can be up to 150 micrometers. So size determines also to some extent what species we are talking about, in addition to the spines.
Also, the presence of putting a cover slip, no, Sarma, with
Yeah.
plasticine.
Yeah. So when you compress the animal, then very difficult to identify again. So you take a cover slip and little bit of plasticine on the four sides of the cover slip and make as a platform and then put that over that so that the object of the microscope does not touch the specimen, but at the same time you can study the features, the morphological features of the individuals. So any other participant has any photograph that we can try to identify? If you have,
Hello, sir. Sir, these photographs are taken from our MSc students during their study. These are all living organism
ah,
in living conditions. We just put the cover slip on the water drop and then take the photographs through microscope in computer or from the mobile phone.
oh, excellent. So next time, whenever you are collecting with the students, please fix the samples in formalin, and then they will be useful for future identification.
Okay, okay. Thank you, sir.
Dr. Nandini, do you have any suggestion for them for anything?
Excuse me, sir.
She
Please.
has a question.
Okay.
Sir, please share that protocol that you shared in the slide about
Yeah.
rotifer fixation and sample collection.
Okay. Yeah.
Thank you, sir. Thank you so much.
I will send the slides to Dr. Das. You can collect from her.
Yes, sir. Thank you so much.
I don't know if you can hear me. Can you hear me?
Yes, ma'am.
I just wanted to suggest, since we are also here tomorrow, if the
Yeah.
students are interested, they can go back to their lab, take a few pictures of fixed specimens of other rotifer species. And if they want to show Dr. Sarma tomorrow, they can also do that. But more importantly, especially for young students who are working in this field, please do take this opportunity, take lots of photographs, send it to us, and we can definitely help you to determine the species. And it would be best also if you are in contact with, I don't know, what about BK Sarma? Is he still doing anything?
I think he retired, but continuing, I think.
Okay. So maybe you can find his email address and also you can be in contact with Dr. BK Sarma.
Okay, thank you very much, ma'am. Ma'am, can there is any particular key for the identification of zooplanktons?
There are many keys, and these keys are based on each group. So you will have keys for rotifers, keys for different families of rotifera. So you will have a lot of keys are there, but it is always very difficult to use the keys because you need to know each structure that they are talking about in the key.
Yeah.
So for that reason, please follow the guide that Dr. Sarma said. The COSTE books are very useful, and any other images. And
Okay.
I think BK has done some work, no, Sarma, with
Yeah,
some
many keys are
Indian
available.
books.
Keys are
Indian,
available.
yeah.
Yeah.
So you
Ma'am,
will have, yes.
is there any site for the identification? We can send the photographs like birds and butterfly. They are doing these sites. There is any sites for
There are,
zoo
I'll tell you.
planktons?
No, I'll tell you that there are, if you want to talk about experts who can
Yeah.
identify rotifera,
Yeah.
there are only 5 who are very reliable now, who are working.
Okay.
So there is Bob Wallace, Dr. Sarma, Hendrik Segers, Russell Scheel in Australia, and Christian Jersebek in Austria. So these are the only 5 people in whose identification you can actually say that yes, it is correct. These people are very kind and they will answer your messages. But for that reason, that's why I'm telling you that there is no site as such available. But consider Dr. Sarma as a site for
Oh,
yourself, and
yes,
you
yes.
can you can send
Definitely.
him all the photographs and he will be able to help you a lot.
Okay.
I hope he doesn't disagree with me later.
No, no, ma'am. Actually, my one of my students is doing working on the zooplanktons.
Yes,
So I am asking about the identification. So I will
please
in
send it
contact
to
with
us.
Dr. Sharma sir.
Yes, please be in contact with him. He will surely help you. There's no problem about it at all.
Thank you very much, ma'am.
You're most welcome.
Any questions?
Hello.
Hello.
Good afternoon, everyone. I am pleased to be stand on behalf of our organizing team. It's my privilege to propose a vote of thanks on this occasion. My heart filled with lot of gratitude and respect for our distinguished guest, Speaker Dr. S.S. Sarma, their invaluable time for us to grace this occasion, but also for enlightened us with their commendable talk identification of rotifers from different sites. He also explained about the how to collect the rotifers and observe under the microscope, identification of species under external morphology and trophical morphology. For the identification of rotifers, use the different types of literature, and he also explained the zoo taxa species. Thank you, ma'am. Thank you, sir, for brief knowledge about the rotifers and excellent lectures. Thank you, sir.
We thank you. We thank you.
Thank you very much. Thank you all.
Thank you very much, ma'am.
Thank you. Thank you so much. So tomorrow we will see you now with another 2 talks, now on manuscript preparation
Okay.
and ethics of publishing.
Thank you.
Thank you very much.
Thank you very much.
So we can disconnect now.
Okay. Thank
Thank
you very
you.
much. Well, we will
Bye
meet
bye.
tomorrow.
Thank you.
Bye.
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 Now I request to all the participants, please gather here after the lunch.
Recording stopped.
पहले जाओ तो सही राधा वेस्टिंग हॉल में ना
Dr. Kritika Namde, Assistant Professor of Botany. I welcome to Dr. Pragya Kulkarni ma'am on the behalf of all organizing team. I welcome to ma'am and I introduce Pragya ma'am. Dr. Pragya Kulkarni ma'am is head in Department of Microbiology, Government VYT PG Autonomous College, Dur. Total teaching experience is 31 years and subject expertise in plant pathology, plant biotechnology, microbial biotechnology, microbial genetics, bioinstrumentation and techniques, biostatistics, genetic engineering, and environmental microbiology. She is IQAC coordinator and deputy controller in autonomous examination cell. Ma'am also has many publications, 53 papers, 6 chapters, and 1 patent. She is fellow of Society of Life Science since 2011. She has got Best Teacher Award and by Microbiology Society India. She is State President MBSI. She has total 3 research projects in which 2 from UGC and 1 from CG course. Under the supervision of PhD program, 1 scholar has awarded, 1 submitted, and 5 is going on. Today, ma'am is going to discuss about the protocol of the study of aquatic microflora. Then I welcome to Kulkarni ma'am. Thank you so much.
Okay, good afternoon all. So you are here to study aquatic microflora and I'll be here for 1 more hour with the introduction of aquatic microflora with you. So the actually very first all, I am thankful for Department of Botany and Zoology for giving me opportunity to discuss and to come in front of you and share the information about the aquatic microflora or the microorganisms uh, very important in our life. Basically, as you know, the microorganisms are present everywhere. So exactly aquatic microflora in concern is a very small or very untouched or we can say very unexplored branch. But here and where the aquatic environment is support as the transport medium or the dissemination mode for circulating microorganisms from one place to other. So in course of time, the basic or exact microorganism, we can say the water molds in terms of fungi, they are present in water only and essential for water environment. But other than that, there are so many microorganisms present in water and they disseminate in very place and cause severe epidemic like diseases or severe damages. Despite of this, these microorganisms are basically decomposers. You are knowing the ecosystem. So some organisms, some biological organisms known as producers, some are consumers like we people, and the microorganisms particularly are identified as the decomposers, cycling of nutrients, decomposition of complex organic matter and so on. So the beneficial as well as the harmful parts of microorganism with reference to aquatic environment we are going to discuss here. Next please. So what I already said, they are responsible for nutrient cycling, primary production also sometimes, and the decomposition system. They are also the indicators of water quality of any ecosystem or any environment. Sometimes they are also indicator as climatic change and ecosystem health, because the water bodies are directly indirectly related to the human beings or the also the animal stock also. So directly indirectly they are related to the ecosystem health. Next please. So very few things about the aquatic microorganism. This is general introduction about the things and what microorganisms do in the aquatic environment, what they are responsible for. So these are two types of microorganism. We can say these are the free floating or planktonic organism, or sometimes they are attached to the surface, hard surface or solid surface present near the water bodies. Next please. So apart from the classification, just we can identify these microorganisms, because microorganism is a general term. So just to start with the bacteria and fungi. Next please. Algae, protozoas, or viruses. These are actually known as microorganisms and present because they are present everywhere. So they are directly, indirectly related to the aquatic environments also. So you are having algae, protozoans in different sessions. So today we will discuss about the bacteria and fungi only for some detail. And we'll have the hands-on session on these two organisms also. Next. To start with the aquatic bacteria, these are the functions that aquatic bacteria perform in the aquatic environment or in the environment near their survival. They are basically some useful nitrogen fixing bacteria, nitrifying bacteria, and even denitrifying bacteria. Just depicted some examples. I will list these examples in later on also. Next one, please. Some are sulfur reducing bacteria, some are photosynthetic bacteria, also decomposers and saprophytic bacteria. They are basically known for the decomposition of organic matter, and some are pathogenic bacteria. Pathogenic to the environment, human beings, and the animal related to the aquatic environments also. Next. So these aquatic bacteria mostly functions for the nutrient cycling, primary production of organic after decomposition, energy production, recycling of organic matter, symbiotic relationship they also perform in some cases. They are also responsible for water regulations and bioremediation. The bioremediation is the very key factor nowadays. It is very essentially studied in all these days, and scientists are just related to the organisms, identification of such organisms that is related to the fast and rapid recycling of waste material. Next. The bacteria related to the water quality management is mostly causing the eutrophication when the water bodies are received so many types of or different varieties of organic matter, polluted things, pollution causing things. So they are causing eutrophication, and the pathogenic bacteria, they are causing the diseases and they spread. We are also known for some typhoid-like diseases, cholera-like diseases that bacteria are responsible for. And these are the waterborne diseases basically. Next. So what we do, why the aquatic bacteria are studied, or this is essential to study or know about the aquatic inhabitation of bacterial colonies. They are studied for the water quality monitoring. They are also studied for aquaculture. When the fish culture or other animals rearing and culturing is done, so the water is the basic environment, and the water may contain some kind of microorganism that may harm to the such fishes or other materials. So aquaculture studies is also important to study the aquatic bacteria. They are also important for bioremediation study that I have already mentioned, and the climatic changes, because they are involved in the nutrient cycling. So they are also related to the climatic changes studies. Next. So the second one is aquatic fungi, commonly known as water molds, but sometimes they are not compulsory or essential water molds. Some other organisms are also associated in their life cycle. They are associated with water bodies or water currents, or sometimes of ponds and stagnant water bodies also. Next. These aquatic fungi are more important in these days because they are having filamentous growth and they required some kind of substratum attachment, and that substratum also get degraded in due course of time, and the reproductive structures may spread through water currents. So back please. So as we classified bacteria on the basis of their role, the aquatic fungi may also be classified as per the aquatic saprophytic fungi. They are mostly responsible for organic matter decomposition. Then aquatic symbiotic fungi, some are aquatic lichens and some are aquatic mycorrhizal fungi. You might be knowing the mycorrhiza. That is zygomycetes members, and they're associated with roots of angiospermic plants. So they are also classified under the aquatic group of fungi. Next one, please. Then aquatic pathogenic fungi that may cause diseases to the plants also, the human beings that consume that water, and the animal that survive in that particular water bodies. So these are some examples. I have listed these examples in the later slides also. Again, what they aquatic fungi do in the aquatic environment, they are also responsible for symbiotic relationship apart from the decomposition and nutrient cycling. They are causing pathogenic, they are pathogenic in nature and causing diseases. Sometimes these aquatic fungi also serve as the food source for the small animals, insects that survive near the water bodies, that survive inside the water bodies. Small fishes are also there, and these fungal groups are serve as the food source of these organisms. Next. The fungal members are not always true aquatic in nature, but they are adapted to survive in the aquatic environment. Basically, the microorganisms need food and space for their survival. This is the thumb rule of basic availability of microorganism in everywhere, anywhere. You just say that bacteria or fungi present in the air because they are having small in size and they transport or disseminate through the air currents. But their sustainability in the aerial environment, their sustainability in the aquatic or any other soil environment also. So when the bacteria or fungi present in any environment, means that particular species or organism is getting space for their reproduction, and that is also getting food, nutrition for their survival. So they are very adaptive in nature, and whenever they get the space and food, they just proliferate or they just survive. Because of they having their motile spores, they are some aquatic group of members, your fungal members, they are having the flagellated bodies, swim cells, that is known as zoospores, the motile spores. That's why they are also easy to survive in the aquatic environment. They are also adapted for low oxygen, so they may survive in the deeper layers of water. That area having low oxygen, dissolved oxygen in the water currents. Again, sometimes they are not true aquatic fungi, but they are adapted for moisture or aquatic environment. And sometimes they produce some resistant kind of fruiting bodies, fruiting structures that survive in long period of time. They just disseminate through the water currents. And whenever they get the solid surface or whenever they get the less water as it is not required for their growth, so such fruiting bodies are the resistant bodies, and they may survive for a long period of time. So these are the adaptation of aquatic fungi for survival in the general fungi also for survival in the aquatic environments. Next, please. The ecological significance, similarly, the disease, we can just study these aquatic fungi or fungal groups present in the aquatic environment for knowing about the prediction of epidemics like things. So disease control, again, just like the bacteria, we can, if we are having the understanding of such fungal members in the aquatic body, we can control the aquaculture environment. We can use these aquatic molds for bioremediation studies also. One thing is, just I have to mention this because these aquatic members or fungal members are having a huge amount of extracellular enzyme production, and that enzyme is just released throughout the biomass, and they are good remediators or fast degradation of organic matter. They are also conserved for aquatic biodiversity. I had already mentioned that the true aquatic molds, true aquatic fungi are very less identified and very less studied in during these days. So there is need for such young scientists who work on these aquatic microorganisms for conservation and identification of such biodiversity. And usually they are water quality indicators. Their presence or absence indicate some kind of quality of water, whether the water is potable for drinking, safe for drinking, or it is used for any other things, or what kind of pollution. We can identify the type of pollution in terms of presence or absence of such fungal members. Just depicted some challenges in future research in aquatic biology for the sake of you participants, because these organisms are difficult in cultivation, limited knowledge of these organisms, and ecological complexities associated with them. Some organisms are not true aquatic in nature, they sometimes survive. So the identification and demarcation is very difficult. For these are the challenging parts that you might face, or you just challenge, you take challenge of these studies. So it is very difficult to cultivate in some cases, and the knowledge is very limited in literature. Next. These are some environmental factors they may associate when you are going to study these such organisms. You may just mention the exact temperature for their growth, the amount of salinity, because aquatic environment is not only the fresh water, they may associate it with the saline water or the marine ecosystems, and they are identified in such cases also. And the amount of oxygen available for their growth, availability of light, and the pollution that is received in that particular aquatic environment. So the study may mention all these factors to find out the growth of microorganism. Next. Now to start with the study, how we go for the study of aquatic microorganism, how we identify, how we can detect these kind of microorganism. You might be having some protocol with you, and you have already isolated or identified some microorganisms. So basically, we have to have the microscope with us, because without microscope we can't even see the morphological characters. But some are macroscopic characters, they may be used as key factor for their identification. Then in nature they are present in many ways, but when we turn them into the laboratory, then collecting the sample in the laboratory, we have to culture them. We have to prepare some specific growth media, indicator media, or selective media for their growth, so that they can be come into the cultural forms. Some molecular biology techniques are also there for quick identification of such microorganisms. Similarly, nowadays the metagenomic studies are emerged as a new branch, and metagenomic support without culturing. The step number 2 is not at all required for metagenomic studies, and directly we can isolate such organism from the source. We need not to culture, we need not to make culture media and wait for the incubation also. And the flow cytometry that allows them to the classification or just like filtering in the shape and size according to, and then after we go for the even direct molecular biological studies, or we can go for the metagenomic studies. So these are the actual methods. Basic thing is there, we have to collect the sample, and the sample is processed accordingly, and isolation of bacteria and fungi is monitored as per the protocol. And when we get the culture, bacteria is cultured within 24 or 48 hours, and fungi may require 3 to 5 days for growth. And after their growth, just we have to prepare slide of such culture or such organism and observe under microscope. And there are microscopic identification key on the basis of morphological characters, and they are classified or identified on the basis of these morphological characters. Next, please. A complete study of aquatic microorganism or any kind of microorganism need this type of listing, in which you have to have them some introduction about what these organisms are known, what these organisms, where these organisms may available, and then we have to identify the method of sample collection. If the water body is very big, we have to collect, means the students are directed to collect samples in 4 or 5 places and just mix them. Or the water source is very small or very limited water is available, you have to collect only one sample, it is sufficient. Then we can directly observe such water sample for such some large type of organisms. Mostly algal or planktonic organisms are observed by this way. But apart from this, bacteria and fungi are mostly cultured. And one method, media preparation, just culturing. Fungi may isolated by the seed bait technique. Oil-based seeds, sesame seeds, yeah, ground nuts, or sometimes mustard seeds are just partially boiled and just deep in the water sample. After a few days, 2 or 5 days, the fungal members may grow. The fungal propagules are there in the water sample, polluted water sample, and the substrate or the bait is, when dipped in the water sample, the fungal members may survive and they start growing. I show the pictures. When you are having such cultures in your hand and then microscopic studies, just list out the characters that you have seen in the microscope. And finally, you can analyze their the list. The water sample is having 5 number of or 6 number of organisms you have isolated. So number of organism you have isolate, mention that organisms, mention their name, and discuss your result with other person, and the report may be submitted in the case of particular one sample that you have collected for study. So this is the general complete report means work task for students that is starting the aquatic microorganism study. Next. To start with sample collection, just simple sample bottles may be used for water collection, or there are some special type of bottles are available that is having lid with them. So the water may be collected from rivers, lake, ponds, or sometimes if available, the saline water, the marine water may be collected. Ensure that the collected containers are sterile. Initially, there is not having any contamination, and then label the containers. Next, please. These are media preparation that I have already mentioned. Some specific media should be there for fungus and bacteria also. Even within the bacteria and within the fungi, there are 2-3 types of selective media that is to be prepared. These are the selective media for growth of specific type of microorganism. Say the EMB agar is mostly used for the E. coli culturing of E. coli. E. coli is the common contaminant of water bodies that is near to the municipal waste dumping, or sometimes the tube well or the borings are associated with the contamination of fecal material. So these are generally indicator if the E. coli is having in the EMP agar media, the water is strictly not used for drinking purpose, or it is not potable for human use. Similarly, the malt extract agar media and subarrowed dextrose agar media that is used for fungal culturing is very common for identification of or isolation of many types of aquatic fungi. And the water agar media that I have mentioned, that seed baiting technique, that's oil-based seeds are partially boiled and keep in the water agar media that is not having any kind of nutrient, only the seeds, organic matter is just attracted by the media, and the water agar is added for the solidification of media. So these are common media mostly used for the isolation purpose of microorganisms. Next, please. When the sample is more polluted, we just predict the sample may having so many types of organism with them, and they may overlap or overcrowd your culture. So it has to be diluted for 10 to the power minus 3 or 4 dilution. There may be a dilution series. We have to pipette the water sample and mix it to the sterile distilled water, and that the serial dilution is used for. This is particularly for bacterial sample, and streak plate and spread plate method is used for inoculation of such water sample. Next, the bacteria is stored for incubated at 37 degrees for 24 to 48 hours, and the fungi is stored up to 15 to 18 degrees, sometimes 30 degree for 3 to 4 days. These are the colony morphological points that have to be observed without microscope. The size, shape, color, and elevation, texture and edge of the bacterial colony, as well as the color, texture, shape, size, and edge of the fungal colony also. Next. For microscopic examination, very first thing is to observe bacteria under gram staining technique. So gram staining is a very initial and very differential method of identification, just to classify bacteria on the basis of gram positive or gram negative characters. This is the indication when the bacteria is having the pathogenic in nature. So we have to take the sample we have to handle the sample very carefully. So initially just we have to mention that the bacteria is having gram positive or gram negative type. And further we go for the biochemical studies. For fungal cultures, we have to prepare a specific slide that I'll demonstrate you in the later session. And the slide, when the slide is very finely prepared, because the fungal cultures are very overlapping to each other, and we have to identify the fungal culture on the basis of their reproductive structures particularly. The pattern of conidia arrangement, the pattern of spore arrangement, the type of fruiting structure, that is more identified. For that, the bacterial, the fungal slides should be prepared very carefully. Next. These are the list of biochemical tests that has to be performed in the laboratory for identification of bacteria. Particularly, these tests are the responsible for some biochemical behavior of bacteria. So production of catalase, oxidation of some sugars, and then citrate production, citrate utilization, sugar fermentation, and indole production or urease test. These are just essential or important tests for identification. On that basis, the bacteria should be classified on the positive or negative tests of biochemical behavior. Next. When we observe the fungal slides, we have to go for observation of hyphal structure. The hyphae is septate or non-septate. The true aquatic fungi are belong to the lower fungi group. and basically they are cenocytic, non-septate in characteristics. So this is very initial indicator, and they are very thin wall cells. So the stain is not direct in dark, and not attached to the structures directly. They may present any kind of rhizoid or any kind of fruiting bodies in the microscopic structures. If there is pigmentation or any color production in the margins of the culture and reproductive structures, that is also seen in the microscopic observation. Next. And finally, the observation recorded is depicted in the tabular form, data form, and with the help of reference books and with the help of pictorial guides, you just identify the particular genera or the species of organism, and finally the reporting is to be done. Next. Just I mentioned the molecular biological studies. We have to go for extraction of DNA, and extracted DNA is subjected to PCR amplification, and then amplified PCR material is just identified on the basis of database available in so many just database sources of there. Or sometimes particular identification is done on the basis of 16S rRNA extraction, and the amplification again through PCR and identification with gene bank. Next. So start with some pictorial structures, then we proceed to the lab for further microscopic examination. These are the list of common bacteria that may found in the water sample. Sometimes they cause severe diseases also. They are also responsible for bad odor in the water bodies. They are also responsible for decomposition of organic matter, and sometimes the the Azotobacter and Pseudomonas are mostly used for the nutrient recycling. They are associated with nitrogen fixation also. So the common organism they are associated with water bodies are listed here. Next. These are some pictures. What we see Azotobacter in the culture, if we culture that, mostly Jensen's and Okan's media is there, the specific media for isolation of Azotobacter and the culture of Azotobacter. So selected, what the use of selected media, they are specifically grow the typical or particular that organism also. The unwanted organisms or the general organisms may not appear in such media. In microscope, when we go for the gram staining, we can see this kind of structures that have to be elaborate later with the help of particular guide, and then undergo the biochemical studies. Pseudomonas species is very common, and so many species are there. Maximum species are decomposers in nature. They are widely used for bioremediation studies, but sometimes they may cause such type of skin diseases in humans also. They are mostly associated with soil or digging the water tanks and the water bodies or ponds also they are associated. So sometimes it may lead to the human skin infection, mycosis like bacterial infections. The lower picture is also the gram staining slide of Pseudomonas species. Next. Then here is E. coli on EMB agar, particular metallic sheen, green and shining colonies are there. Salmonella appear on the blood agar medium, sometimes on chocolate agar media. They appear like this type of colonies. Cryptosporidium appear mostly single isolated colonies, sporadic colonies in the media. Next, please. Legionella species appear light in dark background. Again, chocolate agar media mostly used. Chocolate is not added with the chocolate. That is the burnt sugar thing is there. Then Shigella is generally produced shining halo around the colony growth. They produce some metabolite. They are indicated the decrease of pH, and the media color is changed around the growing colonies. Then general coliform bacteria, they are also present in the water bodies in huge number. Other than E. coli, other enterobacter species or other coliform bacteria may present and they may pollute the water in terms of various characters. Next. Some Helicobacter pylori is also associated with water bodies. Vibrio cholerae and Vibrio parahelmin mollitrichus. These are responsible for cholera diseases, waterborne diseases in the human. They may appear like this. These are the specific media. You may see this, the color of media vary according to the organisms. So each and every organism needs some selective nutrient, selective environment, selective type of indicator for their growth, and they are appear in such colonies. Next. This is just diagrammatic representation. When a common media is used for isolation of Proteus, Salmonella, E. coli, and Shigella, how we can identify the different species in a common plate. So the Proteus species appear in the colony around a halo, just a colorless zone around the Proteus colony, you can say, and colony is having a dark point with center. So we can say this is as nucleated colony. बीच में एक dark आपने spot देखा होगा. So the Proteus appear like this. Salmonella appear comparatively dark in the environment, in the surrounding. E. coli appear, they just grasp the indicator blue, red color media, red color ka stain, and the Shigella appear dispersed colonies, irregular shape of colonies along with two different colors. So this is diagrammatic representation. This is not the true Petri plate. Next. So coming to the fungal species associated with water bodies, mostly common fungal, waterborne fungal species. That is Saprolegnia, Eclia. These two are true aquatic molds, aquatic fungi. And some other are opportunists. They are also survive in the water bodies or near the water bodies. The organic matter is there. They may survive, and the water, they use water current for their dissemination, spread. Next. The upper one is the isolation of Saprolegnia by the seed bait technique. We just added the, here we are using the wheat seeds for isolation of Saprolegnia. The second one is Eclia, that is causing diseases in the fish body. The aquatic system is having Eclia species in huge amount, and that they cause the diseases on the body surface of fishes. Third one is the Allomyces species. They are just survive in the water for some period of time, and when they get solid substratum in the form of plant twigs or wooden parts, they just appear on their surface on a group of colony-like structures. And the lower side is the microscopic examination, microscopic structures of these three, Saprolegnia, Eclia, and Allomyces species. Next, please. You may be familiar with the penicillium, what it looks like in the culture media. The upper side is penicillium, fusarium, and cladosporium. Most commonly, these fungus may be isolated from soil samples also, and they may be associated with the water bodies. Lower side is the structures, they are appear in the microscopic, and that particular difference in their structures, morphological examination, they may be clearly identified as their penicillium, fusarium, or cladosporium species. Next one, please. This is rhodoterula, appear like bacterial structures. And Candida, this is also a fungus, but appear like bacterial colonies that is having single cell or chain of cells. They are not having the filamentous structures with them, and they may appear like bacterial colonies in the culture medium. Next one is the Nigraspora. This is also very opportunist fungus and highly responsible for decomposition of wooden material, or a fast decomposer near the organic matter available in the water bodies, and they are fast growing fungus. Lower even is the microscopic examination on the basis of these characters. Just like you can say the single cells, east like budding, repeatedly budding cells are there. These are Rhodoterula and Candida species, and the Nigrospora is having dark color fruiting structures, dark color ka Spoconidia. Next one, please. These are also common fungus. They are present around soil samples or air current also. They may be associated with plant surface and the water bodies. Aspergillus niger, you may found this as most notorious organism. Whenever you just go for culturing some other fungus or other bacteria, the Aspergillus niger come and appear like a guest. So Aspergillus flavus is associated with stored food materials also. You can just identify this Aspergillus flavus in the household groundnut store seeds के साथ में associate होता है. And just it is also associated with the water currents and water bodies. Next one is mucor. This is also fast growing fungus and associated with the organic matter decomposed. Most of them mucor may appear when the uh, when the क्या कहते हैं वो उसमें हाँ गणेश पूजा और उस समय जब वो सारा विसर्जन होता है ना, बहुत सारे फूल पत्ती जितने भी चढ़ाते हैं, उस समय ये बहुत ह्यूज अमाउंट में ये वाटर बॉडीज में ये अपीयर होता है. And that may cause some kind of irritation, allergy, and skin burning symptoms on the natives live around that. They use that water bodies for their routine household activities. So these are the very fast growing fungal members, Aspergillus and mucor species. Next. Okay. So we may discuss all these things, whatever you get in your culture, we just go and observe the things and the method of identification, preparation of slide, proper slide for identification, staining for particular fungal structures and observation under microscope. Okay, thank you very much. If any doubt or any kind of queries is there, you are most welcome. कि कुछ पूछना है, लैब में जाना है. If you have already cultured or done or go through this type of studies, any kind of difficulty you may face during your studies, you may ask. से है ये फिर. Okay. Or otherwise you have to start with this background. The introduction is, if well enough, you may start your studies in this background and you go just proceed your studies in this direction also. Okay, thank you. Thank you all.
Good afternoon, everyone. I am Dr. Diksha Khare, Department of Botany, to be stand on behalf of organizing team with my privilege to propose a vote of thanks. On this occasion, my heart filled with lot of gratitude and respect for our distinguished guest, Dr. Pragya Kulkarni. Their invaluable time for us to grace this occasion, but also for enlightened us with their commendable talk, study of aquatic microflora. Ma'am, very good lecture and brief knowledge about the fungi and microfungi, aquatic fungi, and there are so many aquatic fungi like microorganisms which are present in water like bacteria, fungi, and different type of water microorganisms. And she also explained the aquatic bacteria, ecological role of aquatic bacteria and fungi, and their applications also. And aquatic fungi is also known as the water mold, and they are the role of in saprophytic and symbiotic nature in our environment. And aquatic fungi is also a source of food. She explained a different type of role of fungi, and environmental factor influence the microflora also. And she explained the study of aquatic microorganism. First, how to collect the microorganism, collection, microscopic study, analysis, and discussions, and brief knowledge about the Aspergillus, Nigrospora, and Mucor. Thank you, ma'am, for this excellent lecture and brief knowledge. Thank you, ma'am. Go to the botany lab for the practical.
Now session is offline. Now the session is offline.
Now this session is offline. The practical is conducted in practical lab, botany lab. Let's go to botany lab.
Thank you very much. Now the online session is over. We are now conducting the practical session in our botany lab. It is offline for the students present here, participants present here. So goodbye. We will meet at 11 a.m. tomorrow. Goodbye.